Live from the North Carolina Museum of Natural Sciences in beautiful downtown Raleigh. It's the Science Cafe with your host, Chris Smith.
All right, everybody. Let's get started. Good evening, everyone. Welcome to the Science Cafe here at the North Carolina Museum of Natural Sciences. Look at all these fabulous people. Thank you so much for coming out to Brain Awareness Night. This is our fourth annual Brain Awareness Night here at the museum. We have lots of great partners, especially with some of the folks at NC State that come out. How many of you were able to take in some of the activities on the first floor over here? Yeah, some really cool and interesting research going on out there, right? Did anybody see the any of the short lightning talks too? Catch any of the small things going on? A few folks. You're nodding and smiling, so they must have gone really well. Yeah, thanks for coming out, taking in all the activities tonight. Here at the museum, we host the Science Cafe here in the Daily Planet Cafe every Thursday at 7 o'clock. Cool and interesting topics relevant to your daily life. We try to do that every week, so I hope all of you will come out and visit with us again. Check out naturalsciences.org for upcoming topics. We've got a lot of interesting things coming up for the remainder of 2019. But tonight we're going to talk a little bit about the brain. Who has one? Yeah, I hope so, although I have some family. That's a terrible joke, isn't it? Although I was looking at the title for this evening's talk, How Your Parents' Stress Became Your Brain's Problem. And those things, right, they sort of go together. Like, I finally have someone I can blame for all of my problems. And if you're here at the Science Cafe every Thursday, you know they are numerous. Many stresses. They didn't laugh at that one. Okay. No repeat. Yeah, now nobody's going to come back. Well, tonight we have a very special guest brought to us. We have Dr. Tracy Bale from the University of Maryland. She's professor of pharmacology and psychiatry and the director for the Center for Epigenetic Research in Child Health and Brain Development. Please welcome to the stage our very special guest, Dr. Tracy Bale.
Thank you. Thank you so much. Thank you all for coming inside. I know I live in Baltimore, and I know you've had rain and bad weather for as long as we have, so it's a beautiful, it was a fantastic day. I flew in this morning, and so thank you for coming inside to listen to and learn about the brain. As we all have a brain, I find that oftentimes as a neuroscientist, you can't hear me? Oftentimes as a I'm very loud. Really. Oftentimes, as a neuroscientist, if I am boarding a plane or sitting next to somebody in a public setting, and they oftentimes will say, "Oh, so what do you do?" I oftentimes hesitate, because if I say, "I'm a neuroscientist," "Oh, that's really fascinating. What do you study?" "I study stress." "Ah, well, I'm stressed. You should study me." Hands down. "I'm stressed. You should study me." So I think because I get that response, most people sitting here tonight, I hope, are thinking the same thing. We have lots of stress in our environment. Stress is very pervasive. And as I am the director for the Center for Examining Child Health and Brain Development, one of the things that we're particularly interested in is not just stress and how it affects your brain, but how it affects your child's brain, how it affected your germ cells before they became your child, and how it'll affect your grandchildren as well. So those are things that we think a lot about. And so on today's topic, first of all, I also want to say, because it hasn't been mentioned yet, and this is a Cafe Science Talk, does anybody out there under the age of 10 know what day today is? Anyone? Pi Day. So happy Pi Day. Do you know why it's Pi Day? No? Anyone closer to the age of 10 who knows Pi Day? Do you know why it's Pi Day? And how many digits can you go out with that? That's okay. All right, we got to 4. That's good. I remember my son when he was in, it's like maybe 10th grade, 11th grade, they challenged each other and he won, he could go out 20, 30, whatever digits. So, okay. So today we're going to talk about your brain. Mental health is something that is a great concern in our current environment. I put some just basic statistics on the slide that you can read there. I think it wouldn't surprise many of you as an educated community to think about the fact that it is so pervasive today, the levels of depression and anxiety, that children very young, so I think one of the statistics is that 50% of chronic mental illnesses start by the age of 14, 75% start by the age of 24. So what that means for our children, I know as the parent, my child's now 19, but I know when he was in elementary school, if you went in and talked to the school nurse or principal and found out the number of children now that are being treated for things like anxiety, depression, and other psychiatric illnesses is quite surprising. So one of the things that we're very interested in the research that I do, which I do a lot of translational research. I do research at the basic level with the mouse being our animal model because we can do mechanistic studies on a mouse. And we can come back to that. Why do you study a mouse? Because they won't let us manipulate the human. So we have to do it in another mammalian animal. I'll come back to that in the end. But we're very interested in understanding how pervasive stress in our environment, and as we all know, it's growing, how that impacts how your brain functions and how important that is across the lifespan. We have young children in the room. We have people who are maybe closer to retirement age in the room. Your brain is constantly changing as it goes across all of those developmental stages and maturation points, and that's very important. So thinking about the brain, how we understand mental health, if you were to take the National Institutes of Health and divide it by the different institutes and look at the progress across disease treatment. So let's start with cardiovascular disease. We've made tremendous improvements in longevity of life. If you look at cancer, we've made amazing in the last decade contributions, longevity of life. If you look at mental health, we have really not. So ketamine treatment was just approved by the FDA. That has been the first drug really with a different target in over 50 years. We have not really made great strides in mental health. It is something that I am a huge advocate for, so if anybody has any interest in advocacy, this is a huge point of we need more funding for mental health treatment. Okay, your brain. The brain is fascinating. Is there anybody here with a brain who does not think the brain is the most fascinating organ in the body? Right, nobody. Stop. It is the most fascinating organ in the body. It is because it's the most complex. It is far and away the most complex organ in the body, and it orchestrates. You can get a liver transplant, you can get a heart transplant, you can get a kidney transplant. You cannot get a brain transplant. It is a very complex organ. It is critical for everything in our daily lives, and it's incredibly fascinating. But how did your brain, as you're sitting here today listening to me, and how it's functioning, how you cope, right? So there's this new idea that has come out of a lot of research that, has anybody heard this, the difference between orchids and dandelions? This is a new concept, and it's really true, right? You either are likely to be on the spectrum of being a dandelion, and what do we know about dandelions? Okay, we call them weeds, but they're also incredibly durable, right? They're very resilient. They're hard to get rid of. They show up in the craziest places, a sidewalk where it's complete concrete, and there'll be a dandelion. And then on the other side, you have orchids. Orchids are beautiful. They are sensitive. They're very pretty, but they're not dandelions. They are not resilient, and they're very sensitive to changes in the environment. I like to say that as our environment in this country has gotten more and more complicated, that we are largely a country that was born out of and developed by dandelions. And if you look at difficulties we're having in generations today, we're raising a lot of orchids. And there's lots of reasons for that, and we can come back to that topic as the dandelion parent of an orchid. We'll come back to that. Okay, so how did your brain get to where it is today? How did you, and the decisions that you make, how adaptive you are, the diseases that you are at risk for, the things that you're resilient to, how did that happen? How did we get there? Well, a lot of early life experiences, many of you have probably heard of these, are called adverse childhood events, or ACEs. These were developed by Kaiser Permanente, studies they did in California. And the reason for doing the adverse childhood events scale was to try and create ideas, and they developed 10 questions they can ask you and that can determine a lot about your disease risk and resilience across your life. And of course, Kaiser Permanente was interested in doing this as an insurance, right, to understand health, what factors contributed the greatest amount. And these are factors, like I said, there are 10 criteria. You can look them up. I'm not going to name all 10 of them, but they have things to do with as you were growing up as a child prior to the age of 18, let's say, did you experience the divorce of your parents? Was anybody in your household incarcerated? Did somebody immediately close to you die? Those sorts of things. Was there violence in your household? Were you sexually molested? There are 10 of these criteria. And what I'm showing you here, I don't know if this is going to work, probably not, but if you look at on the left of the slide there, you can see that if you look at the number of these adverse childhood events, or ACEs, that accumulate over time prior to the age of 18, they're incredibly predictive for disease risk, especially neuropsychiatric disease, if you've had 0, 1, 2, 3, all the way up to 10 of them. Right? So the increased risk for the accumulation of this adversity in your household is quite predictive for your risk across your lifespan to present with a neuropsychiatric disease, which comes back to dandelion and orchid. And so we like to think of this as gene by environment. So you inherited genes from your mom and dad, and I will highlight you cannot outrun your genes. I don't care how healthy you live. I don't care how you get up early and you go for a run and you only eat lettuce. If you have a cardiovascular disease risk, you can't outrun your genes. You can live within the confines to live longer, of course, because you can contribute to that risk, and that's your environment. But you are your genes largely. The gene by environment piece has to do with what experiences and exposures, such as ACEs, you have been exposed to as your brain is growing and developing that put you at risk for things like schizophrenia, depression, anxiety, ADHD, right? It's a genetic risk multiplied times that adversity in your life, right? We understand more and more because of the GWAS studies that have been done, largely funded by the National Institute of Mental Health, what a lot of those gene risks are, but they only predict really for neuropsychiatric disease a very small percentage of your risk. Everything else really is the environment. And we're now understanding more and more about ACEs, those adverse childhood events up until 18, but there's also things that happen during pregnancy and things that happen even before you were a wink in your mom and dad's eye. Okay, so it just shows you the risk on the right. That's what that slide is there. All right, so ACEs, the accumulation of those ACEs. So on the left side of the slide here are studying your at-risk populations. How do we study this? How do we study this in research by looking at things like mice and rats and other animal models? And how do we study it in a translational potential in humans? Okay, on the left shows you the accumulation of ACEs prior to the age of 18 for the US general population. So as you can see, I'm looking at the slide now, so there is about 12.5% of the US population looking generally has 4 or more ACEs prior to the age of 18. And if you actually look at those who have 0, it's 36%. And again, that's going to change depending on where you are in this country. Okay, so I live in Baltimore. Baltimore is a high-risk population. There's a lot of interurban stress that goes on. And so actually, if you look at a population of pregnant moms that were studied and the level of 4 or more ACEs, now you're up to, 61% on that slide? So it's very population specific. So if I'm interested in how adversity, trauma, stress experience impacts a child's brain and how it impacts them across their lifespan, I need to appreciate that if I'm studying a population in Baltimore, those pregnant moms, they have a lot of adversity that they're dealing with from their childhood that affects how they respond to their environment, that affects everything about how their developing fetus is growing. Okay. So because those are the things that I'm interested in, I told you that I'm a neuroscientist, but really, because I'm very interested in all of these factors, I have to appreciate everything about mom's pregnancy. I have to appreciate her uterus and germ cells before she was even pregnant. I have to appreciate dad's experiences and his germ cells at pregnancy. I have to appreciate the in utero environment, everything from that placenta and all the other factors that contribute to how that pregnancy is going to progress. So I call myself a neuroendoreproplacental germ cell androgynecologist. You can look at my business card. Okay. So how does the brain get to where it is, and how do we think about our brains, our children's brains, our parents' brains, and all of that we've had to deal and cope with? And so we have to do a little step back in time if we're going to talk about that preconception environment and visit back to the days of the birds and the bees. Okay. So before you were really a sperm and an egg, right? So before you became the individual that you are, you have sperm from your dad and egg from your mom. And those germ cells didn't arrive at fertilization without experiences of their own. And I'm not just talking about the reproductive tract, although that is important. I'm actually talking about the life and experiences that your dad had. Was your dad in the military? Was your dad a Holocaust survivor? Was your dad really happy and engaged and had a very easy life? Was your mom molested when she was a child? All of those factors contribute to the environment that those eggs and that sperm are then being developed and maturing in. And yes, the maturation of an egg as a germ cell is very different than the maturation of a sperm because they turn over, right? The sperm is constantly turning over, and we'll kind of come back to that topic. But nonetheless, all of the tissues we call the somatic cells that give rise to the turnover of dad's germ cells themselves have experienced over dad's lifetime, right? So it's going to contribute to all of the signals that that sperm picks up on its trajectory through all those tubules that arrives at that egg. So all of them are very important. Okay, so as was mentioned earlier, and as we know that we all like to contribute all of the negative things about our lives, we like to blame mom and dad, but let's face it, we all like to blame mom, right? Everybody blames mom. My mom didn't this, my mom made me do that, it's all mom's fault. All right, so let's start with mom. So when mom is gestating, when she gestated you in utero, and your somatic cells were developing, your germ cells were developing that gave rise to the children that you have. But in course of that gestational event, right, the fetus is growing and developing. First it's an embryo, it turns into a blastocyst. That blastocyst gives rise to cells that become in part the placenta and become the developing fetus. All of those cells, have a sex, believe it or not, right? The placenta has a sex because it's derived largely out of the embryo, right? So if you are XX or XY, so is your placenta. And that's really important because there are sex differences in utero for risk and resilience, the orchids and the dandelions, for all of the experiences that mom has while she's pregnant. So did your mom have an infection? Did she have the flu? Did she have a cold while she was pregnant? Did your mom have gestational diabetes? Was your mom overweight or underweight while she was pregnant? And of interest to my research, was your mom stressed during pregnancy? Now, pregnancy itself is really stressful, but specifically, was there any traumatic events that happened during pregnancy? There is a lot of epidemiological and basic research evidence showing that when those stresses and perturbations happen during pregnancy can determine a lot about how that developing fetus is affected by factors that happen during pregnancy. So one of the things that I'm particularly interested in is why we have sex differences and disease risk. So why is autism 4 or 5 to 1 boys to girls? Why is depression 2 or 3 to 1 girls to boys? Why does depression largely become sex specific around puberty and adolescence? What is it about the sexually dimorphic brain? And yes, the brain is sexually dimorphic. So it's not binary, it's not black and white. It's on a spectrum. There are regions of your brain important for learning and memory. There are regions of your brain important for limbic function and stress reactions. There's the cortical parts of your brain. All of these parts of your brain are on a spectrum in terms of their size and function. And some of them are larger in females and some of them are larger in males. A lot of that is guided and determined very early in life. And so if we were to take all the women in this room and put them in an fMRI, and take all the men in the room and put this in an fMRI, we're going to find that largely you will fall if you're a woman within a normal range and if you're a male within a normal range. If you are a man with schizophrenia and they put you in an fMRI, many of those brain regions that would normally look like a normal male size, larger and smaller, may actually look on the spectrum actually more close to a normative female. It does not make you female, and it does not mean that being schizophrenic is being more female-like. I want to make sure that's clear. But what it means is, during a period of development where those functions are happening, where that sexually dimorphic brain is developing, it suggests that the factors that may contribute to things like schizophrenia are disrupting the normal process of sexually dimorphic brain development. Okay? It does not mean that schizophrenia is being more female-like. All right. Okay. So in utero, so I'm going to go back to that. In utero, we find that most neurodevelopmental disorders, autism, early onset schizophrenia, ADHD, obsessive compulsive disorder, they're neurodevelopmental because we have evidence that that which contributes to this disease disorder occurs developmentally. It may not present until you're 19 or 22 in the case of schizophrenia, but what happened to your cortex, for instance, was probably something that happened in utero. We don't understand all of the contributing factors. We know some of the genes that contribute to the risk. But why is it that almost all neurodevelopmental disorders have a bias in males, meaning that males are more likely to present with them than females? It is also true, is anybody a pediatrician or obstetrician or work in a NICU? Oftentimes when you speak with them, they will tell you that there are the case where there are more males than female babies in the NICU, and that a perturbation that happens to a pregnant mom is more likely to affect a male than a female. And so it may be that males are more at risk, in this case being the orchids, and females may be more resilient, in this case being the dandelion. We don't really understand, but we're interested in if we can understand what presents in risk and resilience factors, then we can understand more about these diseases. So what is it about the in utero environment? One of the things that my lab studies, and was a recent topic in the New York Times that our research was described, was an article that was focusing on the exciting new research on the placenta. That it used to be thought that this placenta was just this throwaway tissue, that it sort of acted like a sponge, wicking information back and forth between the mom and baby, but it's not. As I said, the placenta is actually a sex-specific tissue because it's derived out of the baby, right? But it actually is an endocrine tissue, meaning it's reacting to changes in mom, and it's producing its own signals that are required for fetal development, especially for fetal brain development. So you can imagine if it's a sex-specific tissue, it can be reacting differently to the same environment. We know this from doing studies in rodents because rodents have litters. So within the same uterus, we can expose mom to stress, right? Mice, not very difficult to stress. They're not hugely intelligent creatures. Very easy to stress them during pregnancy. And then you can look at the brain development of her offspring. And so within the same uterus, a male developing and a female developing will respond very differently when we're stressing mom. The males being really at risk and the females showing a lot of resiliency. We've tracked this down to being effects because of what the placenta is delivering in terms of information in that developing brain. And so I'm just putting a little article up here from the New York Times that also showed in the same article was discussing there is a psychiatrist who's actually also in Baltimore, but he's at the Lieber Institute as part of Johns Hopkins, and he actually is a geneticist. And so he's been studying schizophrenia for his entire career. He recently identified loci across 5 different cohorts around the world that contributed significantly to schizophrenia risk. 2 different parts of the genome. Not surprisingly, one aspect, they found a lot of genes important for the brain. Nobody's surprised. What was really surprising is the second region, placental genes, suggesting that, again, this gene by environment. So if you have different SNPs in these regions, which makes your placenta not function the way it should or be too sensitive to changes in mom's environment, it's sending these different signals that affect brain development. And that risk was significantly greater for males than it was for female births. So again, highlighting a lot of this new data showing that we need to understand more about this particular tissue, how it develops, and the signals that it's sending to the developing brain. Okay, So just a little FYI, another area of research that my lab does in understanding how mom contributes to the brain of her baby. You may know, you've heard, I'm sure, a lot about it. I bet there's been cafes about talking about the microbiome. Most of probably what you've heard, maybe the skin microbiome, but probably the gut, right? Everybody's interested in the gut microbiome, right? As you get older, you should know it doesn't really matter how much fat you eat. It's all about the fiber, right? So as you get older, you can go to McDonald's, but you got to go home and eat your bulgur wheat and kale salad for dinner, right? So it's really what matters. So that's the gut. But what turns out also to be interesting is the vaginal microbiome. So for women, the vaginal microbiome. So no matter what, unless you were born C-section, any C-section babies in the room? Okay, we'll come back to you in a second. You're special. If you were vaginally born, as you passed through the birth canal, you ingested a whole bunch of mom's vaginal microbiome, right? So you owe your mom. That vaginal microbiome is really important. It's called the initial inoculant. It's the first bacteria and microbiome, right? The inoculant that your gut sees, you got from mom. And depending on, right, your mom, what she's been exposed to, we'll come back to that. But if mom has been stressed, it changes the composition of that vaginal microbiome. Therefore, the inoculant is different. And so we've done these studies in our mouse model, and we've also done these studies translationally and looking at the humans. And sure enough, if mom has been stressed during pregnancy, it will change the composition of that vaginal microbiome. That baby ingests that. That initial inoculant has a really big job. It educates the gut because it has to build up tolerance with the immune system, right? Because you're going to live together in harmony, you hope, unless you get an infection, but you're going to live together in harmony. And that initial inoculant tells the immune system, based on its composition largely, we're going to be here, it's going to be okay, because the mucosal lining, when you're first born, is not developed yet. So they have a direct interaction. And so it's like, we're going to live here, we're going to help you out, we're going to produce all kinds of key things and nutrients for your brain, we're good. And so if that inoculant is different, and again, to all you C-section babies, if you didn't get it, then your first inoculant probably came largely from the hospital. And that can be, it can be pathogenic in nature, and it's a very different. And they've shown, hands down, that C-section babies are at much greater risk for all kinds of allergies across your lifespan. Right? So that initial inoculant is really important. And so how that vaginal microbiome changes, and this is like a little queasy topic, especially for a lot of men who don't like to think about ingesting their mom's vaginal fluid as they were born, but it's a good thing. In fact, there are currently studies, because we know that the microbiome is so critically important, there are currently studies that are really, so this is showing in our mice. These are cute little mouse pups. So we can C-section deliver them, and we can give them, we can ask how important mom's vaginal microbiome is by inoculating them ourselves and then studying their brain. We're actually currently making humanized mice by taking all the different things we know about women with neuropsychiatric disorders, women who've been trauma exposed, et cetera, and looking at the differences, and then taking that, because a microbiome is a microbiome. The host doesn't matter, and we can inoculate these pups and look at their brain. The studies, actually, the clinical trials that are ongoing in the United States, there's a bunch of them in New York, there's in Canada and Europe, where they actually are taking women who know that they're going to be C-section delivering babies. They take swabs and incubate them in mom's vagina. They take the swabs and they rub them all over the baby when they're born. They orally inoculate them. I don't want to hear any ew. This is all normal. And so they inoculate them, so they're getting that initial inoculant, so they're not getting first exposed from the hospital. And so those are studies that are ongoing, and they're following these kids to see if they can reduce their risk for different diseases and allergies. So very important. Okay, what about dad? All right, so I'm tired as mom, always being the one blamed for everything. All of the translational studies that we do in my lab across hosts of models and stress, and we do a lot of things translationally, looking at autism and schizophrenia and depression and ADHD. We work with a lot of clinics. They will always give us data, because when you walk in that clinic with your child with the various diseases or disorders, they will always ask mom, so what did you do during pregnancy? Did you have an infection? Like, nobody ever says, hey, dad, What happened to you? What were you exposed to across your lifespan? So we've become very interested, as have other groups, in understanding dad's contribution. It's a little bit far removed because you're not gestating the baby, but your germ cells have been exposed, as I said earlier, and that's important. So the United States is hugely fascinated with sperm counts. I don't know why, it's like some mark of masculinity across the country, and our sperm counts are dropping. This is also true in Europe. We have many toxicologists here from the university that study, and this is one of the things, like, what is it about our environment? This may not be politically correct, but I have my theories about the current environment, about what's causing stress. Like them or not, it's a very stressful environment. I think that's reducing sperm counts. So some of the evidence that we have actually goes back to things that many of you have heard of: the Dutch hunger winter, the Swedish famine study. So the Dutch hunger winter was a period towards the end of World War II in which the Nazis caused basically an induced famine in many areas, including Amsterdam and other areas of the Netherlands, in which they induced minimal calories. Food was not accessible. I mean, they were eating tulip bulbs to get calories. And so a lot of studies have been done on women who were pregnant or children that were early developing during this period. And now, of course, we're now many generations out from this period of time. So Audrey Hepburn was a Dutch hunger winter baby, had lots of health problems. There are other famous people as well. And so trying to understand what it was about, I mean, obviously not having enough calories while you're pregnant is also very stressful, I'll point out. But not having enough calories is also a nutrient stressor. So they've studied, and there's a list of things that they've studied of these individuals for their children and their grandchildren. So many generations now showing risk for things like schizophrenia, showing changes in longevity, cognitive function. They've actually started looking at, by taking blood samples from children and grandchildren and trying to understand maybe what the mechanisms are. Changes in immune cell populations are some of the examples on here. So the Swedish famine literature is also full of this. Why do we study dad? Because mom is incredibly complicated. We have mom's uterus, the pregnancy itself, the delivery, her lactation, her behavior. But dad, especially in animal models, we got one cell we need to carry about. One glorious night together with mom, we can take dad away, because in rodents, we don't need to have dad around for the survival of the pups. So we can expose dad to all kinds of things, breed him one night, take him out of the cage, never sees his kids, never knows anything about them. So that's one of the ways that we can ask questions, because it's easier to look at the signals, what we call epigenetic signals, and I'll come back to that in a second. It's easier to do in that kind of a model. Okay, so in trying to understand in dad what it is he can pass on in his germ cell other than his DNA, we call this thing called epigenetics, and I'll come back in a second to what that is. But I'm trying to understand in the sperm what he can transmit, right? He has his DNA, but if it's dad's life experiences, your dad beat you as a child, your mom coddled you as a child, all of those factors influence your somatic cells, those cells that are required for giving rise to spermatogenesis and sperm maturation are very, very important. Sperm themselves are transcriptionally inert. That means a sperm, as it's finishing spermatogenesis and traversing the reproductive tract and is eventually ejaculated, it can't respond to the environment. There's no way for it to transcriptionally change what it's storing or accumulating, right? The cytoplasm is too small. Okay, so what are the mechanisms that we currently study to understand this? Well, we know that Mendel had this idea. Anybody who's gone through school and studied Mendel's peas, wrinkled, smooth, you remember all your Punnett squares from school, right? He understood that the environment could select for traits, and that those traits by genetics were inherited, right? We all understand that. But he didn't account for the influence of an environment. If you had no water available in his garden, did that cause changes that if you then took those seeds, that could be passed on, right? So he didn't account for that. Evolution, you might be thinking, accounts for selection of positive traits, right? Those that are advantageous. But Darwin's theories didn't account for the rapid pace by which those could accumulate, right? Evolution takes too long, right? A good example is our current obesity epidemic. Factors that we can pass on to our offspring. Evolution has not accounted for our obesity epidemic. all evolution cares about is availability of calories, right? Famine. Evolution hasn't caught up with that. Okay, so what about drugs exposure, stress in your environment, infections, and of course, as I said, obesity, right? So how do all of these factors in our environment, how do we pass on those traits to the next next generation or transgenerationally even? How do we account for those changes? Those would be called epigenetics, which I'll explain in a second. Which might, for many of you who remember this from college, sound a little Lamarckian. Lamarckian was a little crazy, and I'm not going to advocate for the fact that he wasn't. He was a little crazy. His idea was, in that draft thing I just showed you, was that while you're gestating, if mom is stretching her neck and getting access to more yummy leaves, there will be this magical signal that will make the neck of her offspring longer. Okay. Biology does not support that, neither does evolution. However, the fact that signals could be passed on and inherited, which is really what epigenetics is, sounds a little Lamarckian, and it may actually be not too far off. And those are things that we study. So what are epigenetics? All right, so epigenetics, if you dig down into your chromatin, into your DNA, epigenetics are all those factors that lie above the DNA, right? So histomodifications, methylation of the DNA, and these small non-coding RNAs, those are what we refer to as epigenetic. Epigenetic lies on top of or surrounds the DNA. The environment is not changing your DNA sequence, because as I said in the beginning, I'm sorry, you cannot outrun your DNA. You can try as hard as you want, you just can't. However, the environment also influences the genes that you inherited from your mom and dad and how they're expressed and where they're expressed and to what levels can be influenced by the environment to more extensive levels. And if those epigenetic marks can be passed on in some way with the germ cells, you can pass them on to the next generation, right? So they can affect your somatic growth, how you are who you are, largely because of mom during pregnancy. But if you passed on effects in your germ cells, those can be intergenerational epigenetic marks, okay? So that is one of the ways, and you can see maybe if you're sitting near a screen, that in sperm, as an example, right? But all germ cells have epigenetic marks in them naturally. They're not special. They're required. They're there. Right? So there is DNA methylation. You've heard of imprinted genes. Those are methylated genes. But the environment can influence other things that are methylated. There are some histones in sperm that are retained. Not many, but there are some. Those can be modified. Right? So they're biochemical marks that are on top of the DNA that do not change the sequence of DNA, but it can influence how things are expressed. And we know that you are here because of gene expression, right? 2 cells became 4, became 8, became a blastocyst. That requires transcription, and that is determined by epigenetic marks, right? Histone modifications, et cetera. Those are all important. There are these other things, though, that are called small non-coding RNAs. So they are RNAs, so they come from genes. They are RNA, but they don't typically code for a protein. They don't have a function beyond that. But they themselves go in and do hosts of really cool things. Largely, these small non-coding RNAs, which we call microRNAs, target specific mRNAs. So if you suddenly accumulate a whole bunch of these things because there's stress in your environment, let's say, they can go in and rapidly, you don't have to change the DNA of the genes that are expressed, you can just attack the mRNAs. And that's largely what these microRNAs do. And there's tons of them in germ cells, especially in sperm. So one of the things is figuring out, because sperm are transcriptionally inert, they're not responding to the environment, how do these microRNA then change with an environment and influence post-fertilization, which is crazy. Well, there's these tiny little vesicles. So if you walk away from this talk with any new information, the word is extracellular vesicles. We call them EVs. Every tissue in your body, every mammal secretes them. Cancer biologists have largely identified them. Tumors secrete them, and so cancer biologists use them as biomarkers. So oftentimes, if you go in and they're looking for a specific kind of cancer or tumor or growth, they can use a lot of these markers now because they travel in circulation. Well, what do? Well, they're teeny, teeny, tiny little lipid rafts. They're they're not cells, but they're lipid membrane-bound tiny vesicles. They have tons of protein in them, and that tells you where they came from and where they're going to. The insides of these things, chock full of microRNA. And it is a way in which, and this might sound a little endocrine to anybody in the audience, right? It's a way in which one tissue can send a signal to another tissue. So you can go from your gonad to your brain. You can go from your placenta to your immune system. And really, frankly, largely, these extracellular vesicles travel in high concentration. It's because it's really tissue's way of signaling to an immune system to let it know sort of the status of various tissues. Pregnancy produces a lot of them to sort of communicate with the immune system so the immune system doesn't attack the developing baby. So these extracellular vesicles communicate with germ cells, and now we have a mechanism. We have a way in which the current environment can change how these vesicles are secreted from cells along the reproductive tract and bring these microRNA in close proximity and attach really with the sperm. And now those sperm can take that signal to fertilization. And that is a communication way in which the environment can influence the course of transcription that happens after fertilization. So one of the ways that my lab has explored this, we developed a way to do what's called stress in the dish. We can take reproductive cells, we can stress them with glucocorticoids, we can collect vesicles, we can look at them in vivo. You don't need to understand this. This is a way of fluorescently labeling these vesicles. And anybody been at the meat market where they have that whole thing where they show you your pig or your cow and where the meat's coming from? I grew up in Minnesota, so we have a lot of this around. And so this is very similar to that for the mouse. We can inject them into the mouse, take out the tissues, and look where those vesicles go. They have an incredible specificity for the reproductive tract. This is one piece of data. If you walk away with any appreciation of data, you can see that on the top are very different colors than on the bottom. And this is just showing you, it's called proteomics. We can collect these vesicles and we can look at all of the proteins. We can sequence all of the protein. And you can see that if you stress a reproductive tract cell, those vesicles carrying those signals, totally different proteins that are important. That's important for how development occurs. And then you could actually do cool studies with animals research that you can then ask translationally by injecting into a zygote all of those differences that you're seeing and asking about the developing brain. And this is a study that we did here, which I'm not going to go through for time, but we can actually ask questions about the developing brain. This is what basic animal research allows us to do, because obviously we can't do that in humans for obvious reasons. But we can stress in the mouse and ask, what effect is this having on the brain? Does this have any of the same gene risks that we think about for autism and schizophrenia and ADHD in the developing brain? And we've actually then taken our studies and asked, I was at the University of Pennsylvania for 15 years, and so we did this study with Penn undergrads over the course of final exam stress, and we paid them, and we collected their semen samples, and we can ask, do we see differences that occur in these individuals? And the answer, in fact, is we do. So we collected their samples from them. And I don't know if you can see on the slide there, on the left-hand side is a very different population of these microRNA in sperm than the right-hand side. Everybody see that? Anybody, right? Very different pattern. So this is from their sperm collected once a month over 6 months, including the stress of final exams. Different men are going to respond differently. And what we found was those that reported stress, believe it or not, you can sequester these two different differences. The no stress guys showed up on one side and the stress guys showed up on the other. So what that tells us is that there are signals. This is really new information. We didn't ask those men then to go out and reproduce. They're too young to be doing that. But we don't know. Of course we don't know what differences are making, but now we can start to ask those questions. We know which changes we're seeing here, which patterns, and then we can go back to our mice again and ask those questions. And that's why basic research is so important. So now that we've talked about mom, I hope we can all appreciate dad, equally important to our developing brain. Don't just blame your mom. So why is your brain so complicated? Why does it function? Again, dandelions and orchids. It's really complicated, right? We don't really understand everything. Dad's germ cells, mom's germ cells, the in utero environment. a lot of influences on the capability, resilience of your brain is not just about because you're tough. It has a lot to do with your mom and your dad, development, and of course all the influences during your childhood. So you can still blame your parents. I'm not an advocate for saying that you can't. Mom and dad are still important. But these are the ultimate factors that we study and why animal model research is so important for understanding that. So I just want to acknowledge before I take questions that this research, of course, is done by trainees in these labs, graduate students, undergraduates, postdocs, as part of their training process. These are just some fun pictures from recent events in the lab, a student's dissertation. And on the bottom there, acknowledging, of course, that the National Institute of Health, we have funding from the National Institute of Mental Health, the National Institute of Child Health and Brain Development, and of course, here in Raleigh, you have, of course, the NIEHS, which we also receive funding for, and trying to understand stress across the lifespan. So we do lots of research in my lab. I talked a lot about brain development. We also study adolescence, pubertal onset, risk over puberty, effects, right? Women with the highest ACEs, so women who've experienced adverse childhood events around and before pubertal onset, the highest risk factor for affective disorders like depression and anxiety across the lifespan, right? Those are our highest predictive models that we have. So we need to understand that. What is it about this unique window? Is this a particularly sensitive time for women? Right? That's middle school. Anybody remember middle school? Really stressful time. So what is it about this window of time and its sensitivity? Right? We have to understand these factors before we can try and therapeutically intervene. Who's at risk? Who are the orchids? Who are the dandelions? So I'm happy now to take any questions.
Let's give Dr. Bale a round of applause. All right. Wow. Okay. Well, yeah, let's take questions. Got a few minutes? I hope you all do too. Hope you've got questions. If you do, wave at me or wave at Katie. She and I have microphones, and we'll bring microphones to you, and we'll get to as many questions as we have time for remaining. But don't yell out because we are broadcasting, and I want everybody in the room, there's a lot of people to be able to hear. We want to make sure we use the microphones tonight. So let's get started.
So this was a wonderful talk and a lot to grapple with. But I'm wondering if you can identify so that we can see how much on the frontier this thinking and research is. Can you identify one thing that you've told us that is not widely accepted in your field, and that's controversial? Yeah, just an example. And also something that you have come upon that you think is important, but that you don't understand, that's a puzzle.
Everything.
Well, you can't get away that
These are
easily.
really, really great questions. And anybody who reads the New York Times knows, somewhere in December, and then they followed up with different articles. One of the big controversies in this field right now is transgenerational epigenetic inheritance. It's a really big. There are our colleagues in my field who feel that they have taken to Twitter. There's like Twitter storms on this topic. I try to stay out of it. And trying to understand, as I pointed out, sperm are transcriptionally inert. Therefore, if you and your wife or spouse are trying to have a baby and you've had some recent trauma experiences, the idea of how those experiences translate into the germ cell and impact post-fertilization into that generation is already kind of crazy. But as I told you, between the Dutch hunger winter and the Swedish famines, there are groups in New York who are studying the Holocaust survivors and the 9/11 survivors and doing multi-generational studies. The controversy in this field is because sperm are transcriptionally inert, and because when sperm and egg come together, the epigenetic marks that I told you about, the DNA methylation, they're all erased. Right? That is a normal process of post-fertilization development. They're all erased. Only the imprinted marks are replaced as the cells start to divide and develop. So there is no mechanism for epigenetic inheritance of DNA methylation across generations. That's the argument in this field. It's a great New York Times article that tries to address possibilities, because what happens is that epidemiologically, people like to take, okay, we can't take biopsies of your brain, right? So if your parents were Holocaust survivors or your grandparents, et cetera, we can't go in and no one's going to offer up their children for brain biopsies, obviously. So what often happens is people will do blood as a surrogate. They'll take white blood cells or other types of cells in the blood, extracellular vesicles or something we're interested in, and they start looking for genes that have been epigenetically modified in groups because your parents were survivors or yours were not. And can they account for differences? And that's where this is where the controversy arises, is that if you're looking at you're a Holocaust survivor or grandchild of a Holocaust survivor, and we see differences in, as I just showed you, there's evidence for differences in methylation of some growth factors, IGF-2 is one of them. How is that possible? Because when sperm met egg, all of those marks were erased. So how are they, right? How do those methylation marks, how are they different? And so it could be you're a Holocaust survivor and you're going to just parent differently. So you're influencing, you're feeding your kids differently, you're disciplining them differently. So those might be factors, right? But then how could it become transgenerational? That is a big controversy in this field. The studies that I was telling you about with those really special extracellular vesicles are one way to sort of get around this argument, in that the vesicles contain marks, microRNA that I told you about. So you're a Holocaust survivor, your extracellular vesicles continue even post you now have left the country, you're a survivor, you're having kids. Those cells, the somatic cells that line your reproductive tract, can continue to produce different signals. It doesn't have to happen at the level of the sperm. At the point of fertilization, those microRNA will produce an effect. So it doesn't have to be methylation. It could be something else, right? So now at 2-cell stage, things are slightly different. Now at the 4-cell stage, they're even more different. And now at the 8-cell stage, right? So you can pass on a signal without it having to be this methylation mark. But that is the controversy in this field, hands down. Yeah, very contentious. Good question.
Back in the back.
Hi.
Uh-oh, baby.
Hello. Yeah, he has a question for you, actually. Just kidding. No, I actually, I'm sure you get this a lot, but I could pick your brain for hours and hours and hours. But one of the questions that I would bring to you is just like your approach or findings in regards to coping strategies for stress, and like how that, like, and also like what are the factors that, because I feel like stress is a growing issue or topic in modern society era. What are the contributing factors to that?
Yeah, and it goes back to this orchid dandelion thing.
Yeah.
And I say it sort of in humor, but it generationally it has become a really big factor. And you know, what's contributed to this? You know, we could have a great conversation all night about helicopter parenting and not letting our kids fall down and letting them learn resilience. And it comes back to kind of those sensitive periods. And I am not judging anybody, because I have a 19-year-old working on some issues. And I think of myself as not a helicopter parent, but, you know, maybe a little bit. If we don't learn resilience factors during critical periods of development, and then we send these kids off to college, that's a different period of brain development. And are those learning those different factors later is harder and different probably, and is that contributing to differences we see in terms of resilience and trying to understand the world around us, on top of the fact that the world around us is really complicated, and millennials are dealing with lots of things that I didn't have to deal with. I did not have social media. I did not have a cell phone as a kid. My parents didn't know where we were ever, and it was a good thing. So it's a different environment today. And so risk and resilience factors, critical important areas of research. We need to understand who's at risk, but we need to really try to understand from those who appear to be incredibly resilient, right? We all know people who grew up in horrible circumstances and just rose above it. I mean, really the biggest kind of dandelions. And we don't really understand a lot about what those factors are, right? No doubt, again, you can't outrun your genetics. And so you won the lottery on the gene pool, even though you didn't win the lottery on environment, but your genes outweighed that environment. And others, You know, they don't win the lottery on genes, and even though you have the best environment, there's some great studies you should look up. Charles Nelson, not the TV guy, but the scientist, studies Romanian orphanage kids, and he gives this fascinating talks. He has all of this data. So he went into Romania. So Romania was at a point in time, they did not have a foster care system. And when the economy was really bad and there was no money available, a lot of parents would put their kids in orphanages. And these orphanages, I mean, they weren't beaten. It wasn't trauma, but it was more on the area of neglect, because they were so full of these kids. They didn't have enough people. So they weren't held. They weren't loved. They had nutrition. They had the basic staples, but they weren't really, right? So there's something about that need, right? There's lots of primate studies that show this as well. You need to know that love, that nurturing. And so his studies showed that if you can get the kids, so he did all these studies, he convinced the government through television and advertising to do studies with foster care. And so he showed that if you could get these kids out of an orphanage by the age of 2, they were totally recovered, totally normal in terms of their ability to bond later in life. They didn't have affective disorders. They weren't at risk. He has these studies where he shows these kids, and he shows a picture of a girl, and he's like, well, how old do you think this girl is? And he's like, yeah, she looks like she's maybe 5 or 6. No, she's 16. Because that lack of nurturing actually is physically stunting these kids. Anyway, so he showed that if you get them out of these orphanages and into foster care in a loving, supportive environment, by 2, you rescue them. After 2, didn't matter. There was something about those really formative years that set the structure. Right? You might be able to rescue different facets and with the right amount of therapy or whatever, but all right, and these studies have shown in the US, lots of kids who've come from, we had friends who had adopted a child from China, I believe, and a similar thing, had been in an orphanage and adopted her, you know, somewhere at the age of 10. She really struggled with social relationships and bonding. And so there's something about the brain early on needing that nurturing to develop that resilience and that the social relationships and understanding and environment. That's really important. Yeah, good question.
Great talk, thank you. In the study on the sperm and the effects of stress,
No, I don't want your sperm.
Did the sperm revert back to normal when the stress ended, and how long did that take?
That is a great question, and the answer is, scarily, it does not. So we actually in our mice, we stress the mice it requires a sufficient amount of stress, right? This is not just one stressor and you pass on an effect. So we found that in our mice, if we do at least 4 weeks of what we call a chronic variable stress, which again for mice is not hard. We just do something different to them every day, a novel object in the cage, change their cage, a different noise, the smell of a predator, something different every day. 4 weeks is sufficient to produce an effect. Anything less than that, not really. Anything more of that you don't need, right? So 4 weeks is enough. If you breed them right after the stress, they don't pass on an effect. If you breed them a month after the stress is over, they do. 3 months, which is a long time in the life of a little mouse, they continue to pass on the same effect to their offspring. So there's something about a sufficiency of information that changes, again, at the level of those somatic cells that are churning out these vesicles. That takes time. It takes time to accumulate. What we haven't been able to figure out is how to reverse it. And I think that's really important, because one of the things we're interested in are like military. A lot of men who serve in the military, exposed to a lot of traumatic events, come home at an age when they're probably thinking about having kids. And if we could develop, so those Penn undergrads, not traumatized, but those Penn undergrads, if we could start to develop a normative sort of prediction of structure, then we can start to ask questions about what looks different. But this worries me, because what if it's lasting, can we figure out reversal? And so maybe if we could figure out predictive markers that put kids at risk, we could figure out ways to intervene, you know, in vitro or some level of that. Sorry.
Hi, I was wondering, do you think that there are actually more stressors in today's society, or is it more just that people are more diagnosed? Because it seems like I have a lot of friends, including myself, who have anxiety and are diagnosed.
Millennial?
Yeah, technically, yeah. And like my parents, I definitely have a family history of anxiety, but no one was ever diagnosed before. We just called it the worry gene. So I always kind of wonder how much is diagnosis and how much is environment.
You've hit upon several truths, which is we have better ability to diagnose people today. More people seek treatment than probably of my generation or your parents' generation for sure. So that's part of it, but it doesn't explain everything. The environment, I would say, I mean, maybe people who are a few years older than I am could speak to whether or not they agree, but I feel like the world today is far more stressful. But I don't know, if we went back to times of the Vietnam War, probably people would say, I mean, I was not alive then, but people would probably, I mean, would you say that the Vietnam War era was a really stressful time? Would you say it was more or less than now? You would say more. I mean, right, so, yeah. Well, some might question today's world, but yeah, right. So I think we've gone through periods of time where the environment is incredibly stressful. I think we hear about it so much more now. Back then, you relied on, you had the news or newspapers, and that was it. But God, now you get this, you're sitting at your computer working, and like the New York Times stuff is just blasting across your screen. It's so stressful. So I think that contributes. I think it's multifactorial, but I do agree, again, having a 19-year-old, and it is not uncommon when I go around the country and do, you know, various things and talk with my colleagues, who we all have kids the same age. It is not uncommon to talk about the struggles of our kids that I don't remember being but you know, we're all selective in our memory. So I think that there are contributing factors, and I think, again, it helps us to try and understand what is different about the world, what is different that contributes to the likelihood of presentation of anxieties and depressions. You know, I was on Penn's campus for 15 years, and it's a stressful environment in terms of the undergrads, right? There's a lot of pressure that they're under. But man, the number of suicides amongst those Penn undergrads, and that you know, is true amongst the Ivies. So, you know, has that increased? The statistics say yes, that has increased. So I think it is particularly worrisome to try and understand stress, for sure.
Do you think there's a limit to how much you'll be able to accurately predict about human health and mental health using primarily translational studies? I mean, is there only so far you can go with that before?
With mice?
Yeah. Or whatever.
Yeah. Yeah, humans are horrible models for mice, unfortunately. Yes, it is difficult, and I struggle with this a lot. Every project in my lab that we do with mice, everything that we're funded to do, preconception male stress, preconception female stress, pregnancy female stress, pubertal female stress, all of our studies have a translational component where I work with clinicians, all of the information that we try to get, and we try to work with things like extracellular vesicles, cell-free mitochondrial DNA, the placenta, because those are translational tissues. One of the things I like about studying stress, as opposed to some other things that you can study, is that the part of the brain that determines how you respond to stress or you respond to stress is identical in my mice. So that's helpful for thinking about how important this is. So if something during pregnancy affects the part of the brain that determines how you're going to react to stress in a mouse, it's highly likely that it's also important in a human. Right? So we see a lot of that translational potential. But you're right, higher order structures, you know, many of my colleagues back there are probably nodding their heads, like higher order structures, how the prefrontal cortex, that last part of the brain to develop and mature in adolescence, as you're growing and developing and going through puberty, that's that last part, right? In men, it's not fully developed probably till 25, maybe 30, 60, 70, somewhere in there. And it's a part of your brain that determines, you know, impulsive control, which is why teenagers tend to be so impulsive, because they haven't put the brake on yet. That part is not really the same in a mouse. They just don't have that cognitive ability. So we struggle. I will say that I just recently served on a panel where the director of the National Institute of Mental Health is kind of doing some pushback on some of these studies, because we use some behavioral assessments in mice, but what do they mean? Some of the things that we do, we don't know. I mean, they're validated, a lot of them are, with drugs that you and I would take for anxiety or depression. That helps us a little bit, but the problem is nobody in this room could be diagnosed for a neuropsychiatric disease unless you come in for an interview. And that requires conversation. And you can't have a conversation with a mouse, unfortunately. They won't tell you how they feel. And that's a limitation to a lot of our studies. But I think, most of my neuroscience colleagues know that, and we take that into consideration. We try to think about what are ethologically relevant behaviors for how stress reactivity in a mouse. So we do a lot of things where they are allowed to hide versus coming out in the open. That's a normal mouse behavior. A stress will encourage them to hide. So there's things that we can study. Mice, despite the fact that they're not very smart creatures, actually you can do learning and memory tests with them, so you can train them, limited way, but you can train them to learn things and ask about learning and memory tasks, right? A hippocampus is a hippocampus. And so there are definite things that many of my colleagues who are standing here that we study that we can translate. Some of it we can't. And so some of that stuff just has to be done. But that's why mental health is so hard, because the brain is really complicated. The human brain especially, very complicated. Yeah.
Hi, thank you for this. This was fascinating. So I have read a lot about this topic for personal reasons, because my parents grew up with a lot of trauma, and their parents grew up with a lot of trauma. I grew up with trauma, and I have a 2-year-old. And so I think about this stuff a lot. I thought about it even before I was pregnant, and then of course during pregnancy. And so my question, I'm always fascinated by this, and I'm always left also like really depressed because I think about like, what do people do who have this, these histories of trauma, and what can I do for my son? And so I'm just curious about the practical implications.
I think by the nature of the fact that you're asking this question means that you're probably already a concerned good mom.
But yeah, but I'm always left with that question of like, where does that leave the people who are impacted by this? Do you think there are, I know you mentioned some studies being done like for the fetus or whatever, but like, what about people who are already here? What do we do with these people?
Yeah. I'm a big advocate of therapy. I don't think there's a person on this planet that couldn't benefit from a little therapy. Right? We all carry burdens with us from things we need to sort of work out, some more than others, right? And those contribute to how we react, how we interact with our spouses and our children and our parents and our colleagues, right? We all carry a little bit of that with us. So therapy is a really good thing. I think therapy has come a long way. Everything from cognitive behavioral therapy, there's this thing called now dialectical behavioral therapy. There's all kinds of new ways, coping strategies. I'm a big fan of exercise. Is a really good therapy. I'm not talking like crazy exercise, but just doing things for yourself, mental health awareness.
Right.
I think in this
more like, what can I do for my kid? Because I know how to take care of myself, but I'm thinking about him and what he's inherited. So I'm curious.
Right.
I don't need to put you on the spot, but
like I said, you can't outrun your DNA.
Yeah.
And I would probably add on to that, you can't really outrun your epigenome as well. But you can, teach them really good, healthy behaviors. You can teach them awareness, right? How you respond to your environment is under your control if you're aware of how you're responding. I think especially for little kids, I remember when my son was younger and I got divorced, and the therapist was a big thing for like, talk about feelings, right? So that you're aware. I think especially for boys, really key. We don't always in our culture encourage boys to be aware of their feelings. Do you talk about your feelings? You're just shaking their heads. Like, don't ask me about feelings. I just think that those things are important, because the more aware you are of your experiences and your risk. My child, there's a huge family history of all kinds of addictions. And so from a very young age, I talk to that, you're at risk. You may think that you're not as a teenager, but you are at risk. So just making them aware without traumatizing them about it. But although for teenagers, it's probably a good thing, you're at risk. I just think that awareness in kids is really important. They need to know things that they need to be aware of and healthy ways of responding to them. I appreciate that you're upset, but hitting somebody, that's not okay. And those kinds of, common sense things, because you can't undo it. I'm going to remind you, I'm PhD, not MD.
How about one more round of applause?
Thank you.
Thank you very much. All right. Things to live by, things to think about on the way home tonight and for the next week until we get to the next Science Cafe. Thank you all for coming out to Brain Awareness Night here at the museum. Thanks to all of the folks who came out and exhibited and shared their research in the Daily Planet Theater. If you're all still in the room, many thanks. Let's give those folks a round of applause and all the work that they did to put on a great event. Thanks to Dr. Tracy Bale. And I hope that we'll see you here again next Thursday night. I hope I got this right. We've got Dr. Scott Belcher from NC State. We're going to be talking about what's in your drinking water. Yeah, you want to be here for that one too. That's 7 o'clock next Thursday. Bye, everybody.